Good evening, students. So, in the last class, we discussed about gene regulation. You can say regulation of gene expression. So, then we studied that gene regulation of gene expression can be done at a various level in case of eukaryotes. Whereas in case of prokaryotes, regulation of gene expression can be done only at the transcription level. And even we know that for our syllabus, they have given only regulation of gene expression in case of prokaryotes. And to understand regulation of gene expression in case of prokaryotes, we should discuss this Lac-operon concept. So what is the meaning of operon? As I said in the last class, operon, it is a polycistronic structural gene. It is polycistronic structural gene present in the prokaryotes and which is having common promoter and operand. There are many types of operon like Lac-operon, tryptophan operon, arginine operon, valine operon. Like that, there are various operon are present in prokaryotic cell. But for our syllabus, they have given only this Lac-operon. So what is the reason for this operon to call as Lac-operon? Here, Lac stands for lactose. Okay. And since this operon helps in a metabolism of lactose, it is called as Lac-operon. Again, question comes, what is the meaning of metabolism of lactose? See, this lactose is a disaccharide molecule, which is made up of two monosaccharides. So one is glucose and another one is galactose. So this lactose made up of glucose and galactose is a source of energy for the E. coli bacteria. Commonly in a E. coli bacteria, the source of energy is glucose. But when glucose is not available, lactose is available. Now this E. coli bacteria uses the lactose as an energy source, and to convert this lactose into energy, they follow this Lac-operon model. This concept was first proposed by two scientists. One is Jacob and another Monod. Jacob and Monod. Okay, these are the two fellows who explained about Lac-operon concept in the year of 1961. In case, year is 1961. So they explained this Lac-operon in a bacteria called E. coli bacteria. Okay, it is E. coli bacteria. And as you can see here in the diagram, Lac-operon can be explained in two instances. One, switch off condition, and another one is a switch on condition. So what is the meaning of this switch off and a switch on? We will be discussing in our today's class. But before that, let me tell you some of the important parts in Lac-operon, right? Or in a operon. See, it is having the various regions. Lac-operon is having various regions. What they do and what they stands for, let us see. First here I have written P. So here P means promoter. And you know what is the meaning of a promoter when discussing this transcription concept. Promoter is a region where polymerase enzyme bind and transcription starts. When a transcription starts and ends, at the end of transcription, we get a mRNA. That is called as a transcription. And in a transcription, there are three steps: initiation, elongation, and termination. And this initiation is done at which region? It is promoter region. And here I, what this I stands for, I stands for inhibitory gene. Inhibitory gene. Or you can say it has another name, regulator gene. So in our NCERT book, they have given a name as regulatory gene or a regulator gene. Okay, that is about IG. Now let us see O. So what this O stands for? O means operator gene. O stands for operator gene. And there are three structural genes, Z, Y, A. Okay, so there are three structural genes. One is Z, Y, and A. See, this Z gene produce an enzyme. Name of the enzyme is beta galactosidase. So what is the function of this Lac-operon gene? Lac-operon gene, it is helpful for the production of beta galactosidase enzyme. This is the name of enzyme. Y gene, it produces another enzyme, permease. Right? It is second enzyme, permease. And finally, A gene produces trans-acetylase enzyme. Trans-acetylase enzyme. Beta-galactosidase enzyme, permease enzyme, trans-acetylase enzyme are the three enzymes produced by ZYA. And students, one more thing is, this is ZYA are called as the structural genes. What they are called as? They are called as structural genes. Okay. So what are the example for structural genes in case of Lac-operon? Lac Z, Y, and A. And here I have written name of the enzymes that are produced by Z, Y, A. Now, why these three enzymes are required? What is the importance of these three enzymes? These three enzymes are helpful for the metabolism of lactose. Okay. What is the meaning of metabolism of lactose? As I said already, lactose is a disaccharide molecule which breaks into two monosaccharides, that is the glucose and galactose. So breakdown of lactose into glucose and galactose is called as the lactose metabolism, and that is done by these three enzymes. And each of the enzymes, they have their own specific function. Now you see beta galactosidase enzyme. This beta galactosidase enzyme, it is the main enzyme that breaks lactose into glucose and galactose. Okay. So what is the function of beta galactosidase enzyme? Beta galactosidase enzyme, it is split, and you can say breakdown of lactose into glucose and galactose. And it has got another function. In our NCERT book, they have given only one function of this beta galactosidase enzyme. But in addition to this function, it also does second function. What is that? This beta galactosidase enzyme converts glucose into, converts this lactose into, not glucose, lactose into allolactose. Okay. So that is the second function of beta galactosidase, conversion of lactose into allolactose. So these are the two functions of beta galactosidase. Now you take the permease. What is the name of, what is the function of this permease enzyme? The name itself indicates function. Permease. Permease means it is responsible for permeability. So what is the meaning of permeability? Say there is a bacterial culture. Bacterial culture means a group of colony of bacteria. These all are think E. coli bacteria. These all are E. coli bacteria. And in a E. coli bacteria culture, if I add a lactose, if lactose is added, say it is a lactose, now this lactose molecule should enter into E. coli cell. Then only it can be used as energy source. Got my point? Lactose, when it is added to the bacterial culture, that a lactose should enter into the bacterial cell. Entry of a lactose or any other molecule into the cell by crossing plasma membrane is called as a permeability, that you studied in first year syllabus. Permeable, impermeable, selectively permeable. So same here also, the entry of a glucose into the E. coli bacteria, that is called as a permeability, that is the function of permease. In a simple way, I can say permease is responsible for uptake of lactose into the E. coli bacteria. Now you take trans-acetylase enzyme. This trans-acetylase enzyme, what it will do, it is responsible for a reaction. Name of the reaction is trans-acetylation. It do or to perform a function that is called as a trans-acetylation. So students, there are three enzymes involved in a Lac-operon, and all three enzymes, they have got their own function, specific function. Beta-galactosidase, helpful for the breakdown of lactose into glucose and galactose. Permease enzyme, it is responsible for intake of lactose into the E. coli cell. Trans-acetylase enzyme, it performs a chemical reaction called as the trans-acetylation. Now the question comes, in a E. coli, whether these three enzymes are produced all the time. Will a E. coli bacteria produce these three enzymes all the time, or whether it produces only in a particular situation? See, in a E. coli bacteria, these three enzymes are produced only when a lactose is available for E. coli bacteria. Now, bacteria is getting sufficient lactose. Now, it want to convert this lactose into glucose and galactose. To do so, it needs these three enzymes, and hence three enzymes are produced. Just imagine there is no lactose in the medium. There is no availability of lactose. When there is no availability of lactose, what is the use of these three enzymes? Even they are produced, just imagine they are produced, it is of no use. That's why these three enzymes are not going to produce when there is no lactose medium. So one thing is clear. What is that? In the last class, I said you two types of genes. One is constitutive genes and another one is called as regulated genes. Regulated genes means these genes are expressed only at a particular situation, particular condition. And what is another type of gene? Constitutive genes. These genes express all the time. But here we can say these three genes, Z, Y, A, they are regulated genes. Okay. Now, here I have written two terms. One is called as a switch off, and another one is a switch on. So, what is the meaning of a switch on and a switch off? When these three particular genes produce these three enzymes, that is called as the switch on condition. When these three genes do not produce any enzyme, that is called as the switch off. You can call it in another way. When these three genes are expressed means they successfully produce these genes. When these three genes successfully express or produce enzymes, it is called as the switch on. When these three genes do not produce any enzyme, that is called as the switch off. It is called as the switch off. When enzymes are produced, switch on. When enzymes are not produced, switch off. So how it occurs, let us see with the help of diagram clearly. First, we will discuss about switch off condition of Lac-operon. So, just imagine there is no lactose. Lactose is absent. There is no lactose. When there is no lactose, now these three genes should not produce enzymes. Okay, they should be prevented from expression. And they should be prevented from producing enzymes. How it occurs? In an absence of lactose, this regulatory gene, I already said you, I stands for regulatory gene, or you can say it is also called as inhibitor gene. This regulatory gene or inhibitory gene, it undergoes transcription. And what happens after transcription? mRNA is produced. So this is the mRNA. Okay. This is the mRNA of regulatory gene. Now again this mRNA undergo translation. You know that mRNA is used for translation process where the information present in mRNA is used for synthesis of a particular protein. That process we call as translation. So same thing happens here. mRNA produce a protein. And name of that protein is repressor protein. So what is the name of the protein? The name of the protein is repressor protein. So it will be like this. Repressor protein. Now what happens? This repressor protein binds to the operator. So what is the meaning of O? O stands for operator. So it means our repressor protein binds to the operator. Means this operator is blocked now. When an operator is blocked, why? These three genes first produce mRNA. That mRNA is used for synthesis of protein. That protein will be converted into three different enzymes. So in a simple manner, to produce three enzymes by these three genes, there should be transcription and there should be translation. Transcription occurs from promoter region. Now here, at this place, polymerase enzyme will bind. Polymerase enzyme bind. When polymerase enzyme bind, it cannot pass through the operator. Why this polymerase enzyme, DNA-dependent RNA polymerase enzyme, why it cannot pass through the operator? Because operator is blocked. It is blocked by what? It is blocked by repressor protein. Hence, DNA-dependent RNA polymerase enzyme cannot pass through operator. It cannot reach ZYAG. When polymerase enzyme cannot reach ZAYG, there is no mRNA. When there is no mRNA, no protein. When there is no protein, there is no enzymes because enzymes are made up of proteins. This condition is called as the switch off condition. So why here the term of switch off is used? Because here these three genes are completely prevented from expressing. Okay, they are not producing any kind of enzymes here. Okay, and how it is achieved? It is achieved, silencing of these three genes is achieved by blocking of operator. Operator is like a channel, it is like an entry, right? It is like a tunnel. Through this only polymerase enzyme should pass. It starts, polymerase enzyme always starts transcription from promoter region. But here, since the operator is blocked by repressor protein, now it cannot reach ZYA, hence there is no enzymes. This is called as a switch off condition. It is called as a switch off condition. And students, when it occurs in a E. coli when there is no lactose. Just imagine, there is lactose. It is second case. When lactose is present, now let us see what happens. in presence of lactose. This is in absence of lactose or no lactose, there in presence of lactose. Now what happens? Even when a lactose is present, this regulatory gene, I stands for regulatory gene, it produces mRNA. Okay? This regulatory gene produces mRNA. Now this mRNA become protein. You already know what is the name of that protein. That protein is the repressor protein. Okay, so there will be repressor protein. This is the repressor protein. Now actually what happens? Before repressor protein binds to the operator, instead of binding with the repressor protein, instead of binding with the operator, it will bind with lactose, because here lactose is present. Say it is a lactose. Right? Now, when repressor protein and a lactose bind, this repressor protein becomes completely inactive. Now there is a formation of complex. Okay, inactive complex. This is how it is formed. This is inactive complex is formed by binding of repressor with the lactose. Now this repressor protein become inactive. Once repressor protein become inactive, it cannot bind to the operator operator gene. Now operator gene is free. When operator gene is free, DNA dependent RNA polymerase enzyme bind to the promoter. Now easily it can pass through operator because operator is open now. When polymerase enzyme reaches Z, Y, A, they produce mRNA, right? They produce Z, Y, A, they produce mRNA. This is the mRNA of Z, Y, A. Okay, this is the mRNA of Z, Y, A. You just compare both the diagram. Whether in first case, is there any production of mRNA by Z, Y, A? No. Why here mRNA is not produced by Z, Y, A? Because here no transcription. Why transcription not occurred here? Because operator is blocked by repressor. Then why here transcription is occurring? Because operator is free. How come operator is free here? Why it is not blocked? Because operator is mainly blocked by repressor protein, but now repressor protein has become inactive. How it got inactive? It got inactive by binding with the lactose. Here no lactose, that's why repressor protein is still active. But here lactose is present, that's why it got inactive. Now, these three ZYA produce mRNA. Now, this mRNA undergo translation. When it undergo translation to produce a protein, that proteins are converted into three different enzymes. What are the three different enzymes? We already discussed it. Z produce beta galactosidase. What does Y produce? Permease. And what this A produce? Trans acetylase. It means Z, Y, A, they have expressed themselves in the form of enzymes, but here they were unable to express. That's why it is called as the switch off condition, and here it is called as the switch on. Now a question comes. Okay, fine. Three enzymes are produced, that is beta galactosidase, permease, transacetylase, all are successfully produced. What is the use? What is the use of these three enzymes now? As we discussed at the beginning of this class, beta galactosidase helpful for the breakdown of lactose into glucose plus galactose. That is the function of this beta-galactosidase enzyme. Permease, its function is to allow entry of lactose into the bacterial cell. That what we call it as permeability. Then what is the function of transacetylase? Transacetylase helpful for the acetylation, the type of chemical reaction acetylation. Now all the three genes are successfully expressed. It is called as the switch on condition. It is called as the switch off condition. And students, can you identify what makes a Lac-operon to switch off and switch on? Who will decide whether this operon should be switch on or switch off? Who is responsible for that? It is presence and absence of lactose. Am I right? When a lactose is absent, switch off. When a lactose is present, switch on condition. So here, lactose is like a inducer. So what is the meaning of inducer? As I said in a previous class, inducer is a chemical substance that induces gene expression, that helps a gene to express. So that's why, since here lactose is responsible for expression of these genes, hence lactose is called as inducer molecule. Even in our NCERT book, they have given in presence of inducer and in an absence of inducer. Here, for understanding purpose, I have written absence of lactose or no lactose, and presence of lactose. Here I have written both are same. Inducer, which is the inducer in case of Lac-operon, it is the lactose. And one more thing that we should try to understand is, I say that repressor protein binds to the lactose molecule. Actually, lactose and a repressor bind, and this lactose will bind to the repressor protein in the form of allolactose. Allolactose is one form of lactose. Okay. So this allolactose is one kind of lactose molecule. It will bind to the repressor. So you may expect a question like repressor protein during Lac-operon, repressor protein binds to the which molecule? It is allolactose. By chance, if allolactose is not available, lactose is available, that the lactose will be converted into allolactose by this enzyme. I said no, it has got two functions. One is breakdown of lactose molecule, another one is conversion of lactose into allolactose. Once allolactose is produced, it will bind to the repressor protein. When a lactose and a repressor protein binds, this repressor protein undergoes structural changes. Repressor protein undergoes structural changes when it binds to the allolactose. After undergoing structural changes, it cannot bind to the operator. It has lost its ability. It has lost its ability to bind with the operator. It cannot bind. And this repressor protein, it has got another name. It is also called as allosteric. Right? Repressor protein is also called as allosteric repressor protein. So what is the need of calling this repressor protein as allosteric repressor protein? Because it undergoes its structural changes. Right? It undergoes its structural changes when it binds with the allolactose. Whenever any chemical substance changes its structure upon binding with another chemical, right? So whenever any chemical changes its structure upon binding with another chemical, such chemicals are called as the allosteric. So students, this is all about the Lac-operon. And what is the purpose of this Lac-operon? The purpose of Lac-operon is very simple. This is a best explanation to understand how genes are being regulated. Right? So here gene is being regulated. How this operon is being regulated? When lactose is present, it is expressing. When lactose is not present, it is not expressing. So who is deciding here? The gene expression is under the control of lactose. Sometimes gene expression may be under the control of some chemicals. Sometimes gene expression may be under the control of environment also. Okay. So inducers may be different. In this case, lactose is the inducer. And this was explained by us. We know that Jacob and Monod, they got Nobel Prize for this successful explanation of lactose. Right? So now let us see what are the possible questions that can be asked from this Lac-operon concept. Okay. So one way of asking question is very simple. One way of asking question is they will give a diagram of this Lac-operon. They will give just a diagram of Lac-operon, and after giving this, they will ask you to identify here A, B, C. So what are the three enzymes produced by Z, Y, E? Right? That is Z is producing A means here beta galactosidase, permease, transcriptase. So it is one way of asking question. Another way of asking question is, recently in Karnataka CAT also they asked one question. Just look at this diagram. This Lac-operon is having many regions. Promoter regions, regulatory gene, or it is also called as what? Inhibitory gene, promoter again, operator, lactate, Y gene, A gene. Out of all these genes, can you identify the gene which is constitutive gene? What is the meaning of constitutive gene? It should express all the time. Okay. So here any such a gene is present in a Lac-operon? Yes. There is one constitutive gene. Which is that a constitutive gene? Yes. Inhibitory gene, or what is another name of this inhibitory gene? This inhibitory gene is also called as regulatory gene. And students, a regulatory gene is called as a constitutive. Why? Because you just compare these two diagrams. When the lactose was absent, still it produced repressor protein, means it expressed it. When lactose is present, then also it produces repressor protein. It means presence or absence of the lactose doesn't bother this gene, IG. It expresses in both cases. It means it is expressing all the time. That's why regulatory gene is called as the constitutive gene. It is another way of asking question. And one more question you can expect here. By looking at the Lac-operon diagram, can you predict where the gene regulation is or gene expression is regulated? See, once upon last class I said you that regulation of gene expression can be done at a various level. That is during transcription level, or during transfer of mRNA from nucleus to the cytoplasm level, or during post-transcription changes level, or during translation level. So just recollect all the things and try to answer this question. Where here gene regulation is done? Is it translation level or transcription level or post-transcription level? Correct answer is, in case of Lac-operon, regulation of gene expression is done at transcription level. You may ask me, sir, why it is? Because we already know that in case of prokaryotes, gene regulation can be done at only one stage, that is transcription. That's why in our NCERT book also they have given regulation of genes in case of prokaryote is predominantly means mainly, it is done predominantly at transcription level. So here Lac-operon, where the gene is regulated at the transcription level. So please do remember these all things about Lac-operon. Okay. And in addition to these all things, in addition to these all different structure, there is another gene there is another structure called CAP. So what is the meaning of this CAP? Catabolic activator protein. It is catabolic activator protein, right? So what does the CAP stands for? CAP stands for catabolic activator protein. What is the meaning of this? Very simple. This is responsible for successful operon, Lac-operon. And what is the function of this cap? It has positive impact on Lac-operon. Positive means it is helpful for the expression of gene, not to suppress the gene. That's why it is called as a positive effect. So how it is going to play its a positive role in case of Lac-operon? Very simple. Cap region is present here in a promoter, in promoter region. See, you don't get confused. There are two promoters. One promoter is for regulatory gene. One promoter is for these three genes. All these three genes, they have common promoter. And this is called as a promoter of Lac-operon. And here what happens, a region is present called as the CAP. And what is the use of this cap? This region helpful for the identification of binding the site to the polymerase enzyme, DNA dependent RNA polymerase enzyme. This is the enzyme which produce these three, mRNA from these three genes. And you can say transcription. For transcription, we need a polymerase enzyme. That polymerase enzyme should bind here in a promoter region. And how do it come to know? How do this polymerase enzyme come to know exactly that promoter is here? It identifies promoter, DNA dependent RNA polymerase, I'm saying. That polymerase enzyme exactly identify this promoter region with the help of this cap. So that is all about Lac-operon concept. Okay. Now, this operon, Lac-operon, is called as the inducible operon. Why it is called as the inducible operon? Because in a previous class we discussed that there are two types of operon. One is called as the inducible operon, and another one is called as the repressible operon. Inducible operon, this Lac-operon is inducible. Why? Because its regulation is under the control of inducer. Which is that inducer? Lactose. What about this repressible operon? Example for a repressible operon is tryptophan operon or TRP operon, it is called as. It is an example for repressible operon, right? And that is not there for our syllabus, so you just focus on Lac-operon. So that is all about Lac-operon. So after Lac-operon, what is given for our syllabus? They have given about another concept called as the HGP. So what is the meaning of HGP? Human Genome Project, right? It is HGP. Human Genome Project. So it's abbreviated for this HGP. So students, now only two concepts left. One is HGP. After this, there is another concept called as the DNA fingerprinting. So now these are the only two concepts left. And once these are finished, we can complete this chapter. Okay. Now let us see Human Genome Project. What is the meaning of Human Genome Project? Just recall the thing that you studied at the beginning of this chapter. What we come to know that All organisms, not just human, all organisms, they have got their genetic material, nothing but DNA, commonly DNA. Okay. So this DNA, what it will do, it stores genetic information in the form of genes. Am I right? Now this genetic information is stored in a genes. A question comes, whether all humans are having same genes or different genes. This is the simplest question. Here I am not going to explain much about genes, term gene, because you already studied in detail in the principles of inheritance and a variation chapter. But here my question is very simple, whether all humans share common types of genes or different. Answer is so simple. If every human is having, all the humans are having same genes, then we could have been same, 100% same. But it is not the case. Humans are different from one person to another person. It shows that genes are not the same in all the humans. They are different. Again, a question comes, if the genes are different, how much percentage? How much percentage of genes different from one person to another person? So this question led to begin a project, that is the Human Genome Project. So basically, what is the importance of this Human Genome Project? The main aim of this project is a scientist, they want to study all the genes, and their location, not just genes, their location, their function, and their sequence also. Please do remember, they tried to study sequence of a gene. Sequence of gene means gene is made up of nucleotides like ATGC, and they are arranged in a particular sequence that is called as the gene sequence. So scientists, they wanted to study different genes of organisms, say human, not just a gene, it's a structure, it's a sequence, it's a location, it's a function. These all aims led to start Human Genome Project. By the way, if you compare a human, one person from another person, how much percentage of gene differentiation occurs? All the humans, they are sharing 99.9% of same genes. See, all the humans are sharing 99.9% of same gene. If you compare man and a monkey, particularly chimpanzee, our genome and chimpanzee genome is 99% same. 99. It is just 1% of a genome that made a chimpanzee into man. That is why sometimes, when we will be alone, our ancestral character comes out. Okay. So this is the similarity of 99% of similarity between a human and this chimpanzee genes. But if you compare cow and humans, cows and humans, they share 80% of, more than 80% of genes same. You may feel wonder, our gene and banana genes, banana plant genes, it is 60% same. Right? So we all humans are sharing 99.9% of same genes. If we compare our genes with the chimpanzee, it is 99%. And our DNA and this cow's DNA is 80% same. Our DNA and the banana DNA is 60% same. So this is how the genes are similar. You may ask a question, sir, how it is possible? How it is possible that all organisms are sharing same DNA, same genome? Yes, it is possible. How it is possible? Again, answer is there in this chapter only, genetic code. You know that what is the meaning of genetic code, and you studied already properties of genetic code. And in the properties of a genetic code, what you studied, one property, genetic code is nearly what? Universal. Since the genetic code is nearly universal, almost all organisms are sharing similar DNA or genes. Right? Now, it is another thing, but main purpose of this Human Genome Project is, what is the main purpose of this Human Genome Project? Scientists, they wanted to know, or they wanted to study all the genes that are present in a human cell. Okay? Not just a gene, it's a structure, it's a sequence, it's a function they try to study. And that project they call it as Human Genome Project. Okay. And this Human Genome Project, see here, this term, genome. What is the meaning of genome? So you can define a genome as the sum total of all the genes, or the total amount of DNA in a haploid cell. If you take a haploid cell from a human and study calculated or studied the entire genes and the entire DNA of that haploid cell, then we can call it as genome. Genome means sum total of all the genes or total DNA of a haploid cell. That is called as a genome. So basically, in this project, scientists they studied genome. Okay, that's why they call it as Human Genome Project. And it is not just 1 or 2 years project. It took almost 13 years to study all the genes of human cell. It took almost 13 years. How? It was started in the year of July 1st, 1990 it was started, and it got ended by April 2003. So almost 13 years it took, right? So it's a 13 years project, and it was started, Human Genome Project was started by US Department of Energy. It is a one department of US country, Energy. US Department of Energy. Okay. And National Institute of Health. Same again. USA, National Institute of Health. So these are the two institutions which started a Human Genome Project in the year of 1990. Okay, and later they were supported by some other institutions like Wellcome Trust from London. Right, there is one trust called as the Wellcome Trust in London. It also supported US for this Human Genome Project. Even Japan, China, France. So all these countries, they become major partner of this Human Genome Project. Okay, and you know that what is the main motto or main aim of this project? To study the sequence, to study and to sequence all the genes of humans. Okay, and you may feel wonder that for this project we need samples. We need a sample from different people of different countries. So they collected samples from different people of different countries. They collected DNA from the samples. They conducted a Human Genome Project successfully, and they ended by the year 2003. Okay, and one interesting thing is there is a man called Watson. So there is no need of special introduction about this Watson. You know that. Watson along with the Crick, they proposed one of the most famous successful model of DNA. Right? This Watson was the first person. Watson is the first person who got his genome sequenced. Right? Human genome sequence, Human Genome Project, when it was started, no, his DNA was completely sequenced. He was the first person. Watson. And one more thing is, this HGP or Human Genome Project, it is also called as Mega Project. It is also called as Mega Project. So why it is called as Mega Project? Mega means big. So this HGP is called as a Mega Project because of some reasons. One reason is it took almost 30 years. Another reason is at the end of At the end of this project, whatever the information the scientist got, if they had to return in a books, just imagine they got all the information of this HGP scientist and they want to store this all information in a books. So how many books we need? We need merely 3,300 books. And each book having 1,000 pages. And in each page, you should write 1,000 words. So a book having 1,000 pages, where in each page you can write 1,000 words. Such books we need 3,300 books to write all the information of this HGP. Just imagine how much information one come to know at the end of this human genome project. That's why it's called as mega project. So after this is all about the introduction of HGP. So again, they have given objectives of HGP, goals of HGP, and what are the applications of HGP, and we shall discuss in next class.